Hello guys, welcome back. Today we're doing DNA code of life. Now for you to be able to answer the questions under this topic, you need to know the process of protein synthesis. Before we go through the process of protein synthesis, let's look at what we call a nucleotide. A nucleotide is a building block of nucleic acids. Nucleic acids, we're talking about DNA and RNA. Now, a nucleotide has three parts, which are phosphate group, sugar, and nitrogenous base. A nitrogenous base, which is the most important part in a nucleotide. Now, let's look at the structure of DNA and RNA. A nucleotide is the building block of nucleic acids, which means that DNA and RNA are made up of nucleotides, meaning that on the structure of DNA and RNA, we should have multiple nucleotides. Now, these are the structures of DNA and RNA. As you can see, they are made of multiple nucleotides. Let's talk about the differences in DNA and RNA. DNA is double-stranded and RNA is single-stranded. What does this mean? If you take a look here, DNA has two lines of nucleotides pairing and RNA doesn't. It's just one line. In DNA, the sugar in the nucleotide is deoxyribose sugar, while in RNA it's called ribose sugar. The nitrogenous bases, we have 5 of them. In DNA, you can only find cytosine pairing with guanine and adenine pairing with thymine. As you can see in this picture, G for guanine, it's pairing with C for cytosine, and T for thymine, it's pairing with A for adenine. The bond between the nitrogenous bases is a weak hydrogen bond. In RNA, I will show you when we go through the process of protein synthesis how it forms. Now let's look at a small history of DNA. DNA is double helix, or it's helical. This is the shape of DNA. If they ask you who took a DNA picture, your answer should be Rosalind Franklin. And if they ask you who discovered the structure of DNA, if it is 2 marks, write James Watson and Francis Crick. If it is 1 mark, just choose between the two. DNA can be found in the nucleus, mitochondrion, and chloroplast. These are the locations of DNA, and these are the possible questions you can find in an exam. Now let's look at the process of protein synthesis. Protein synthesis is a process whereby protein is formed. It has 2 processes, which are transcription and translation. The first process, transcription, takes place in the nucleus, and it says that the double helix unwinds, the weak hydrogen bonds break, the DNA molecule unzips to form 2 separate strands. One strand is used as a template to form RNA using free nucleotides from the nucleoplasm. The mRNA is complementary to the DNA, and the coded message for the protein synthesis is thus copied into mRNA. Now the translation says that it takes place in the cytoplasm. Each tRNA carries specific amino acids. When the antigon on the tRNA matches the codon on the mRNA, the tRNA bring the required amino acid for the ribosome to the ribosome, and amino acids become attached by a peptide bond to form the required protein. You might be asked for 5 marks to describe this transcription and translation, and this is what you should write. This is how the process of protein synthesis takes place. Now, when the DNA unwinds, it changes from this shape to this shape. Remember, the 2 strands are pairing through nitrogenous bases. Assuming that we have nitrogenous bases ATA and CTG here on this strand for DNA. Remember, it says that one strand acts as a template to form mRNA, the messenger RNA. This will be the strand that acts like a template. Then it will form mRNA, which will have nitrogenous bases A, UAU from ATA, and you will have GAC from CTG. Why are we having this? We are pairing them. Remember, in RNA, A pairs with U and G always pairs with C. Use this sequence when you're trying to find the base triplets. The base triplets we're talking about UAU. UAU is a base triplet. Now, the base triplets for mRNA are called codons. Now, the mRNA leaves the nucleus and it attaches itself to the organelle in the cytoplasm called ribosome. Take note of that. tRNA, or the transfer RNA, comes with specific amino acid. This is the amino acid, and they have names, but you don't have to know them. The tRNA pairs the mRNA using the corresponding base triplets. Now, the base triplets for tRNA are called anticodon. Then it means that on the anticodon, we will have triplets AUA from UAU, and we'll have CUG from GAC. There is no T in RNA, and the protein is now formed. The bond between the amino acids is called peptide bond. Take note of that as well. Now let's look at the other examples. If you are given AAA, base triplets AAA for DNA, and they want tRNA, you use the same sequence. First, you get the triplets for mRNA, which will be UUU, and for tRNA, it will be AAA. Now, if they give you tRNA, which is ACG, and they want DNA, you use the same sequence. You move from tRNA to mRNA, which will be UGU, and then from mRNA to DNA, it's going to be ACG. Now, let's say that they give you the other side of DNA. Remember, DNA is double-stranded, and they give you TAG. You are not going to use this sequence, but we said that T pairs with A and C pairs with G in DNA. So your answer will be ATC. The sequence will help you to find triplets from DNA to mRNA, from mRNA to tRNA, and vice versa. Thank you so much for watching. If you have questions, please write them down below the comment section, and I will answer